So thank you so much for providing me this opportunity. My name is Charlie. I'm currently an ophthalmology resident at the University of Buffalo, working with Dr. Meg DeAngelis. So what we're looking for is we're trying to leverage GWAS studies of Alzheimer's disease and using pleiotropy to look for overlapping genetic architecture between AMD and Alzheimer's disease. So as people who watch this know, AMD is a very common disease, the most common cause of blindness in industrialized countries. And Alzheimer's disease, similar to AMD, is also a very common cause of dementia, and in fact, the most common cause in industrialized countries. Both of these diseases share a lot of similar features, and both of them, they're very complex genetic disorders where age and family history are the most important risk factors for the disease. They both have vasculopathic risk factors. And finally, in terms of the embryologic origin, the brain and the retina actually are both derived from neuroectoderm. The optic nerve is actually something that sprouts out from the forebrain during development and then helps form the retina eventually. So there's lots of reasons why we believe that there's a lot of overlapping features from these diseases. And this has actually been explored extensively in the literature. There have been people who have done association studies where they look at AMD and Alzheimer's, and they found that people with early AMD have signs of early cognitive decline. People who have later AMD also have a higher rate of biopsy-proven Alzheimer's disease. Similarly, when we look at the cellular levels, we see that both of the diseases, the hallmark features are extracellular deposits. In Alzheimer's disease, the deposit that we talk about is amyloid beta, and in AMD, the deposit we talk about is drusen. And then there have been additional studies when we look at these extracellular deposits that show similar features between the two of them, such as the deposition of complement protein. So with all that background information, what we are hypothesizing is that these two diseases are related in some way through some underlying pathophysiologic pathways, and that we can potentially use leveraging the access to the eye, kind of provide a new methodology to help us look at the risks of Alzheimer's and potentially even develop a method of screening for Alzheimer's disease by looking at the eye itself. With that being said, one of the things that we have to do is first, provide more evidence of the linkage between these two diseases, and that's where the pleiotropy analysis comes into play. So right now, there are many GWAS studies, which are excellent studies, where you kind of take a large population of patients that are either disease positive or disease negative, and using GWAS, you can identify certain genetic loci that are associated with the disease. And this is a great type of analysis in that it's very agnostic and allows you to identify common variants that are associated with the disease. And there's one large study by Frichte et al. 2016 that was published in Nature Genetics, and another one that was on age-related macular degeneration that has, I think, close to 16,000 or 17,000 AMD patients and controls. Similarly, Kunkel et al. Published a large analysis for Alzheimer's disease, which included almost 50,000 patients in the Alzheimer's patients and the controls, where they looked at, did a GWAS analysis for Alzheimer's disease. What we hope to do is to leverage both of these papers and do what's called a multi-trait analysis of GWAS, or MTAG, to look at the pleiotropy between these two. So one of the things I do want to emphasize is the importance of this type of pleiotropy analysis. So traditionally, meta-analyses in GWAS will have one issue where, because, as I mentioned in the Kunkel study, the total number of patients is around 50,000 versus only 16,000, 17,000 in the Frichte et al. Paper, a traditional meta-analysis would weigh the outcomes based off of the total number of patients, which is more of just the number of patients is more so just a representation of how much funding and how much research goes into a particular disease. But instead, what we want to do is a pleiotropy analysis, which puts equal weighting on both of those diseases. And essentially what we want to demonstrate is certain SNPs that are associated with both diseases in a statistically significant way that's greater than the single disease alone. So using this type of analysis, what we did is we performed the MTAG using either Alzheimer's disease or age-related macular degeneration as the primary trait. And with this analysis, we were able to identify several different SNPs, loci, and genes that are associated with each one of these and kind of order them based off of their statistical significance. Using this, we were able to identify several different pathways in both of these and also look at which ones were overlapping. We can find several genes that could have pleiotropic effects on both AMD and Alzheimer's and prioritize them based off of which ones are most likely to be significant. When we do this type of analysis and actually take these genes and run ingenuity pathway analysis, we identified a couple of different pathways, and the most important and statistically significant of these was the complement pathway, which we know has been involved in the AMD field, but now also, again, in the Alzheimer's field, we show that it's something that demonstrates pleiotropy there as well. So one of the very important aspects about the pleiotropy analysis is that not only are these SNPs that we identify significant in AMD and significant in Alzheimer's disease, but they're actually more statistically significant when looking at the two diseases together.
How can these findings translate into patient care?
I think one of the important things that we want to demonstrate here is this underlying disease process between both AMD and Alzheimer's disease. One of the things that's great about the eye and why I love the eye is that we can visualize almost every single aspect of it. And unfortunately for Alzheimer's disease, this is a disease that is actually only clinically diagnosed during, when people are alive, but the true diagnosis is dependent on an autopsy that's done post-mortem. One of the things that will be fantastic in the field of Alzheimer's is if we could really show that there is this underlying pathogenesis between AMD and Alzheimer's disease. There's the theory that we could potentially be screening for Alzheimer's disease while patients are still living and asymptomatic before the disease actually becomes present in these patients. There's actually been some studies out there that show that within the retina itself, and again, I just want to remind you that the brain that leads to Alzheimer's disease and the retina that leads to AMD are embryologically derived from the same tissue. So, if they are both undergoing the same type of process, perhaps we could look at the retina to look for certain changes that might predict Alzheimer's disease. And so far, there have been studies that have already demonstrated some changes within the retinal architecture, looking at the retinal ganglion cells and also the blood vessels that have been associated with future risk of Alzheimer's disease. And there have been some studies that demonstrate that, you can see these changes before the symptoms of Alzheimer's disease become present. Hopefully, in the future, we'll have more data on this, and then we can use this as a screening tool to actually look in the population to see who might develop Alzheimer's disease and potentially even develop therapeutics to prevent the onset of Alzheimer's disease.
So in the future, do you think that Alzheimer's could be identified with a simple ophthalmic exam?
So in the future, I think one of the greatest benefits of ophthalmology is the fact that a lot of the things that we do are very non-invasive. For Alzheimer's disease, prior to the passing of these patients, there are certain tests that you can do, MRIs, which are extremely expensive, CSF analysis to look for tau protein, which involves a lumbar puncture, which again is a very invasive procedure. It's not a very comfortable procedure. I've had to do a couple of these on patients during my intern years. And once you go to ophthalmology, everything is just so elegant and patients are much more comfortable. Sometimes nowadays you have these non-mydriatic cameras where you don't even have to dilate these patients. They just come in, you're able to get a fundus photo of these eyes, and you're able to see so much into their health that may be missed. There are patients who have diabetic retinopathy who don't actually know they have diabetes yet. There are patients who have hypertensive retinopathy who don't know that their blood pressure is uncontrolled. And using these non-invasive ophthalmic tests, it allows the ophthalmologist to act a little bit like a primary care where they're able to screen for these diseases before the patient develops any symptoms of the disease. And looking forward, it's possible that if we can demonstrate, these underpinnings between Alzheimer's disease and AMD, this ophthalmic imaging might also be used in this manner as a screening tool for Alzheimer's disease.
Is there anything you'd like to close with?
I just want to thank Dr. Meg DeAngelis so much for providing me with these opportunities to work with her and in her lab, as well as her mentorship, and of course the AMD Foundation for all of their support. They're the ones who provide the funding to people like us to do this outside the box thinking that I really believe and I really truly believe is going to help push the field forward and lead to new developments that we're all going to be very excited about in the future.
Congratulations on graduating. We look forward to following your career. It has been our privilege to be a part of your journey.